Morning, everyone. First of all, dear professor, and then I would like to say thank you very much because today I can show my presentation in Advanced Reproductive Biology. The seminar is an extraordinary occasion for me to improve my knowledge, especially in the master degree course at the Department of Marine Biology. My name is Angga Saputra. I'm from the Zooplantology Laboratory also. The title of my seminar presentation is The Effect of Overwinter Temperature on the Survival of Female Adult Mud Crab Scilla paramucine under a Recirculating Aquaculture System, as examined by histological analysis of the hepatopancreas and expression of adaptosis-related genes. The material in the paper that I delivered was sourced from a high reputable journal, namely Aquaculture, which was published this year. There are five main points that I will explain, as well as a table of contents in my seminar presentation, which are introductions, material and methods, results, discussion, and conclusion. Introduction. Scilla paramucine is an economically important crustacean of Fortinidae and Scilla. In 2020, its yield in China was 159.43 tons, accounting for 55.45% of China's marine crab aquaculture industry. Although the yield of Scilla paramucine is high, there are still many problems with artificial breeding technology. For example, mud crabs breeding still depends on wild resource, resulting in gameplay instability. One major reason for this dilemma is the lack of artificial cultivar high-quality parent crab, resulting in the shortage of artificial seedling. Therefore, this technology or artificial breeding technology for mud crab is crucial. Zhejiang Province is the northern boundary of the natural distribution of Scilla paramucine in China. The low underwater temperature, which has been kept at 5 until 15 degrees Celsius in winter, it is a challenge for mud crab aquaculture. Generally, crabs are not fit during overwinter for three reasons. The first, crabs feed little at low temperature, and because metabolic rates are low, they usually don't lose much weight due to the fasting. And the second, fasting could save feed costs and help maintaining the water quality of the recirculating aquaculture system. And the last, fasting could stop the spread of the disease with fish like the fresh fish and bevel fish. Then, during overwinter, females are favored. Why? Because the ovary is an important edible part, and then females have a higher survival rate than male calves, whose fitness declines significantly after copulations. Studies have shown that light intensity and salinity affect Scilla paramucine during overwinter, and this system or recirculating aquaculture system. However, temperature plays an even more critical role in overwinter success than light intensity and salinity. Temperature is a critical environmental factor for aquatic animals. Moreover, studies on temperature have analyzed other aspects, including growth and development, stress, immunity, and metabolism also. Nevertheless, few studies have examined the effect on temperature on the crustacean under overwinter conditions. One of the previous studies, the overwinter survival rate of crabs under the constant condition of 15 degrees Celsius and 17 degrees Celsius was higher than that under the natural water temperature, such as 8.6 until 16.9 degrees Celsius. The objective of this study to determine the optimal overwinter temperature range of the mud crab Scilla paramucine by evaluating the survival, antioxidant activity, tissue damage, and expression of HSP-related genes and apoptosis-related genes. And then, the results of this study are expected to improve understanding of physical adaptations of Scilla paramycin to temperature and optimize the overwinter management of mud crabs. Method: Crab source and rearing. Female Scilla paramycin mud crabs were obtained from an aquaculture farm in Taozhou. Taozhou is one of area from Zhejiang province. Before the trial, the crabs were individually cultured in crab recirculating aquaculture system to acclimate for 3 weeks and feed 4 fresh clam for crabs at 9 am. After 3 weeks, 144 healthy crabs were randomly selected and distributed into experimental recirculating aquaculture system. The animals were not fed during the experiment, and 10% of the water were exchanged every day 2 p.m. Water quality is controlled and determined. This is an experimental design. Full water temperature, such as 8, 12, 18, and 20 degrees Celsius, were studied with 4 sets of recirculating aquaculture systems for 8 weeks. The water treatment process at the recirculating aquaculture system is given in this figure. Its recirculating aquaculture system flow rate 2,400 liters consists of 12 blue tanks and the diameter 100 centimeter, height 100 centimeter also, and the volume 700 liters with 3 crabs in each tank. 3 plastic baskets were placed in every tank as shelters to prevent cannibalism. For each treatment, 4 tanks were grouped replicate and were and together with 3 replicates, 12 individual per replicates, and a total of 36 crabs per treatment. There are several data analyzed in this study, namely, first, histology analyzed to gain deeper insight into temperature adaptation and physiological response. Second, reactive oxygen species analyzed to measure the antioxidant capacity. And the third, commercial keys to determine the superoxide dismutase, catalyzed total antioxidant capacity, and malodialdehyde. And 4. Commerce brilliant blue method to measure the total protein content. And 5. Imaging software to analyze 5 randomly selected regions for each section. The gene expression analysis. Among crustaceans, the hepatopancreas play an important role in metabolism and immunity. Therefore, the hepatopancreas was chosen to test the expression of HSP-related genes and autopsy-related genes, wherein, together with 3 replicates, total 6 samples per treatment. Quantitative PCR analyses were conducted on Lake Shaker's 96 rocks to evaluate this gene expression. The table 1 lists the primary used for quantitative PCR in the study. Then, statistical analysis. Before analysis, the Kolmogorov-Smirnov test and Liveris test were used to check the normality and homogeneity of the data respectively. The data conformed to a normal distribution and homogeneity was analyzed by analysis of variance with one-way ANOVA with two-cat multifocal comparison for host test using SPS 2022.0 software to estimate the optimal overwinter temperature for survival. Two models were used to fit the survival agent temperatures such as quadratic and broken line model which line in Excel 2019. Our programming language version 4.0.4 was used to create a heat map to analyze the expression of HSP relations. The data were plotted after the normalization method of contrasting the data, only subtracting the mean without other processing. Other graphs were generated with GraphFed print version 9.0. Results with survival rate. The overwinter temperature had a significant effect on the survival of the mood-scribe Scilla pharmacine. The effect was parabolic with the higher and lower survival rates found at 60 degrees Celsius with 88.9% and 8 degrees Celsius with 22.2% respectively. In addition, the survival rate did not differ significantly between the 12 degrees Celsius, 16 degrees Celsius, and 20 degrees Celsius group. The two models show that the optimal overwinter temperature for the higher survival in woodcraft were 60.1 and 40.2. The histological changes in the hepatopancreas are shown in this figure. In the 16 degrees Celsius and 20 degrees Celsius groups, the structure of hepatopancreas were well organized, the cell boundaries were observed, and the star-shaped lumina were integrated. In the 80 degrees Celsius, the hepatopancreas structure was indistinct and the boundaries were damaged. In addition, the star-shaped lumina, arthrophytes, and hemocyte extracepted. The hepatopancreas of the 12 degrees Celsius group had a relatively clear organizational structure, but the basal lamina was detached from the tubule. Moreover, the cells were highly facultative. Antioxidant capacity. The content or ROS under different overwinter temperatures in this figure. ROS increased with decreased temperature. This content in the craft at 8 degrees Celsius was significantly higher than that in all other crafts. As shown in this figure, the SOD activity of Metcraft exposed to 8 degrees Celsius was significantly lower than those exposed to 16 degrees Celsius and 20 degrees Celsius, but no remarkable difference was observed between the 8 degrees Celsius and 12 degrees Celsius. The CAT activity at the crafts at 8 degrees Celsius was significantly higher than that of all the other groups except 12 degrees Celsius. And then the TAOC in the crafts from the 16 degrees Celsius group was significantly higher than that in crafts from the three other temperature groups. Moreover, the MDI level in crabs were also affected by temperature, and MDA content was significantly higher in 8 degrees Celsius group than the 16 degrees Celsius group. Relay this slide about apoptosis the hepatopancreas cells. The overwintering temperature significantly affect the apoptosis the hepatopancreas cell of the mud crabs. As shown in this figure, with the decrease in temperature, the number of apoptosis cells increased remarkably. Moreover, the apoptosis index of the hepatopancreas in the 8 degrees Celsius group was the higher, which was considerably higher than that the other temperatures. Moreover, the apoptosis index of the hepatopancreatic cells of the crabs didn't differ substantially between 16 degrees Celsius and 20 degrees Celsius group. The relative expression of EHP relation was significantly impacted by the overwinter temperature. With the decrease in the temperature, the relative expression level of EHP 60 and 70 were remarkably up-regulated and significantly higher HV 60 and 70 expression level were observed in 8 degrees Celsius group. Nevertheless, the expression of 4 HV genes like 10, 40, 19, and 78, which were clustered together, were dramatically downregulated in the 8 degrees Celsius group. The Overwinter temperature significantly affects the expression of the V53 gene. The relative expression level of the V53 gene in crabs exposed to 8 degrees Celsius and 12 degrees Celsius were markedly higher than those in crabs exposed to 60 degrees Celsius and 20 degrees Celsius. The relative expression of the BAX gene was significantly upregulated with the gradual decrease in temperature. Nevertheless, no significant difference between the 60 degrees Celsius and 20 degrees Celsius group was observed. Meanwhile, the relative expression of BCL2 and COX4 genes saw a similar trend with the change in temperature, with the highest level observed in crabs at 8 degrees Celsius. The relative expression of BAX or BCL2 at 8 degrees Celsius was significantly lower than that the other three groups. In addition, the relative expression level of Cas3 was also dramatically influenced by the temperature, with the highest level observed in the 8 degrees Celsius group, which was markedly and higher than those in the 60 degrees Celsius and 20 degrees Celsius group. Discussion. Change in ambient temperature affect the survival and physiological process of organisms. In this study, the survival rate of Scilla in 8 degrees Celsius group was significantly reduced, and the optimal overwintering temperature for survival rate of the Scilla was extrapolated to be 14.2 until 16.1 degrees Celsius. However, the previous studies suggest that the overwinter temperature result of the swimming crab was 6 degrees Celsius. Thus, the results indicate that the mute crab has a comparable poor adaptation to low temperature, as a crab is mainly distributed in tropical and subtropical zones. Then, still in the study, the tissue structure of the hepatopancreas in the 8 degrees Celsius and 12 degrees Celsius groups show varying degrees of cell degeneration, necrosis, and structural change. It's worth noting that the loom structure gradually disappeared at the low temperatures. Then, many studies, as the previous research, have examined the effect of low temperatures on physiological and behavioral response, development, and growth of animals. The researchers say that similar in this study, it served to reduce adverse effect during the aquaculture system. This is a figure of possible mechanism of low temperature effect on the survival rate of female Scilla pharmacine. The result indicate that the milk crab has a comparatively for adaptation a long-term contrast and effect might be mediated through the HSV response, antioxidant tissue damage, and apoptosis pathways. In the study, the expression of HSV60 and 70 genes clustered together in the heat map and increased significantly with decreasing temperature. The excessive accumulation of ROS in crabs under low temperatures may cause protein degeneration, which call for the damage the antioxidant capacity. Apoptosis is a procedural cell death regulated by gene and play a vital role in removing damaged and necrotic cells to maintain dynamic equilibrium. In this study, Chun analysis showed that temperature significantly influenced the state of apoptosis and hepatopancreas cell and the mute crabs at egg apoptosis index. Conclusion. To summary, this study discussed the effect of the overwinter temperature on survival rate, antioxidant activity, tissue damage, and stress response genes expression level in the female adult mud crab Scilla pharmacine. Significantly, the optimal water temperature for the survival rate of Scilla pharmacine During overwinter, especially in the street, what extrapolate to be 14.2 until 60.1 degrees Celsius. The result could inform an improved understanding of the mechanism of temperature adaptation and provide guidelines for the overwinter temperature in a much graph in the recirculating aquaculture system. That's all from me. Thank you very much.